welcome to Science Friday.
Here are some for E as well. The one on the top here is one of the ways that you can define E.
What makes a field more likely to have an unbalanced gender distribution?
So this can be, like, in the case of this paper, gyrification or brain folds.
A neuroscientist started to look at the brain
computers,
as
like the
more as an
math
interconnected
are very
incredibly negative
area.
All right, so as you guys might have heard by the various announcements advertising this, I'll be presenting about DNA bank data and COVID and ethics and kind of the intersection between all of those things. So this is a photo I chose just, you guys are all, I'm sure, familiar with 23andMe. This is one of the ad campaigns that they did. It's just kind of showing how prevalent it is, like people are giving it as gifts. It's really popular to do these at-home DNA tests, and I'm sure a lot of you guys have, like, your parents have done them or either you have done them. So your genome is 4 to 5 million SNPs, which is variations in the genome that pretty much dictate everything about you. 4 to 5 million SNPs is pretty much equivalent to 4 to 5 million data points. And with over 12 million 23andMe customers worldwide, and 23andMe is not the only data bank or biobank data place, it's just the largest one. So with over 23andMe, or 12 million 23andMe customers worldwide, that's a lot of data for them to use. So an example of the use of the data is in the paper I'm going to be talking about. But yeah, this raises some questions like, how can this data be used? How is it being used already? What can it tell us? And is it good that my data is being used? Because you have all this data that might be in a biobank, and it can probably tell us some things like this study. So the paper that I'm going to be telling you guys about is, the title is Trans-ancestry Analysis Reveals Genetic and Non-Genetic Associations with COVID-19 Susceptibility and Severity. So pretty much the main thing that this study uses is GWAS, which is a genome-wide association study. It identifies loci associations, which pretty much means it consolidates the data and looks for connections between certain things that are inputted into the GWAS. So this specific paper looks at the test positive versus test negative, aka the infection rates of COVID, as well as 4 different tests to test severity within using the GWAS tool. So this is one of the charts that came from the study, and this is looking at the test positive versus test negative rate using GWAS. The axes on the bottom here, we have chromosome. There's 23, as you guys are familiar with. 23 chromosomes in the human body. 23andMe hence the name. And then the y-axis is negative log to the base 10 of p. Pretty much the higher something is on the y-axis, the stronger or the more robust the association is. So this one is testing for test positive versus test negative rate. So what the GWAS study did is it put all of these data points through, and these are the specific loci or places on the genome where there was seen to be a robust association between the COVID-19 test positive versus test negative rate and this specific place on the genome. So yeah, these two places have the highest and most robust association. And then this one is looking at severity. It's a similar thing with similar axes. So a point here would say there's not much of a correlation, whereas this LZTFL1 gene, or place on the genome, is very strongly associated with severity of COVID. However, this y-axis doesn't tell us if it's a positive or negative association. It simply tells us that there is an association. And then this is taking what we found here.
I have a question. So is each data point an individual and that's a different allele that they're correlating to the severity of the outcome? Like that's a gene, right, of someone's genome that they're associating to the severity of COVID?
Yeah.
Okay.
I don't know what each specific point is, but I just know that along this axis is just each of the points on the genome.
Okay.
Oh, sorry. I'll do that next time. So yeah, if we look back at this one, we see on this loci, which is pretty much an area of the gene, we see an association. And we can look in on a more zoomed in version of this chart. It's called a Manhattan chart, by the way. And we can see this is that same ABO gene that was identified here. And this is more zoomed in so that we can identify the specific SNP that's directly associated with the infection rate of COVID-19. So same axes, it's just more zoomed in, and through this more zoomed in Manhattan chart, we're able to see that the specific SNP that was most associated with infection rates is RS9411376. And then, as I mentioned before, the fact that there is this association, that it's high on the y-axis, doesn't actually tell us if it's a positive or negative association, which is where this comes in. As you can see on the bottom is the OR value, and the OR value will tell us if it's a positive or negative association. What they did here is they split up, these are the African American, Latino, and European are the 3 different ethnic groups that they had the most data on, and they separated those out to make sure that this, or to verify that this robust connection between that SNP and the infection rate was similar across all of these ethnic groups. And what they found is that it was. As we can see, the meta-analysis is like kind of using them all up there. And we can see from this that it is robust, and there is a connection between all of these different ethnic groups because the OR value is below 1. An OR value that's above 1 would indicate a positive association, and an OR value of below 1 would indicate a negative association. So we can see these are all below 1. The meta-analysis is below 1. So that indicates a negative association with this SNP and infection rates of COVID-19, or specifically the test positive rate, meaning people with this SNP are less likely to test positive and therefore indicating a lesser susceptibility to the COVID-19 virus itself. Any questions about that part? Awesome. So what's the bigger picture? How does this connect to everything else, not just COVID? So obviously, the DNA bank data was super important to use for COVID because it's readily available. 23andMe has this data, and we can quickly find out things related to infection rates and severity. But it's not just COVID, and even it's not just disease prevention that this sort of GWAS study can be used in. If you guys think about it, the SNPs, your SNPs in your body dictate almost everything about you. So if we're able to use GWAS and determine the correlation between certain SNPs and certain traits, that can tell us a whole lot about your genome and what you're susceptible to, what traits you're prone to having, things like that. So, I mean, does anyone have any ideas of some SNPs that would be interesting to hear about a connection to, or a trait that you might want to find a correlation between? Yes?
I actually have a question about the previous slide.
Yeah.
So you said that people with this SNP are less likely to test positive, and then you said that indicates less susceptibility to infection. Can you talk a little bit more about that connection? Because it seems like it could be that they're just as susceptible to infection, but that their test result, they don't produce as many antibodies, or whatever they're testing for doesn't show up as well, right?
That's true. So it's not really an exact measurement because this self-reported test positive versus test negative rate is the closest thing that we have to actually showing susceptibility. But you're right, that raises the question of how accurate is this measure of the self-reported thing, because pretty much what happens is all these people have their data in the biobank, being 23andMe, and then they're asked to complete the survey, one of the questions being, have you ever tested positive for COVID-19? So that's, I guess, the measurements that they're using. But it's not super exact, and this translation between this people with this SNP being less likely to test positive isn't super direct to indicating lesser susceptibility, but it's the best measure we have. Yeah?
What does RS94 blah blah, what does it do?
So it's just RS whatever. I don't know that it does anything. It's on the ABO loci, which is your blood type is ABO. So the SNP specifically doesn't do anything except, I guess, make you less susceptible to getting COVID. But it's pretty much just a variation in the genome that some people have, some people don't, and the people who have it are lucky enough to maybe be less susceptible to COVID, or else less likely to test positive. Any other questions? Yes?
Yeah, so I remember actually seeing a genome-wide association study in 2020 that I think identified similar genes, and I think that found that cytokine signaling was a big function of that genes. So I'm just wondering, like, is that like a function of this genome-wide association study also found, or?
Wait, so you're asking about what was the thing?
Is like cytokine signaling also like a correlation that this study found? Like,
I think the correlations that this study were looking for were just the test positive versus test negative rates, and then 4 different tests relating to severity, depending. I think they were just looking at symptoms and stuff like that. But that's like another use of the genome-wide association study for sure. So yeah, going back to talking about like the so what and maybe the ethics behind it, is if this is like a way, something that's readily available, these GWAS studies, and it might be feasible for in the next, 10, 20 years for everyone to have access to their DNA or like biobank data through 23andMe, because if you've done 23andMe, you can go onto the website and look at all your different SNPs. So you could, if you've done a DNA test like 23andMe, go back and look and see, oh, do I have this SNP? And therefore, you're going to find out, are you more or less, or are you less susceptible to testing positive for COVID? That's something that you guys could find out right now. So the implications of knowing that might not be super extreme, but there are certain SNPs or traits that you could find out, or like associations you could find that maybe would be like, have a greater impact on your life. Yes, I see a question.
When you say less or more, like what's kind of the sense of scale there? Like is it like a percentage, or like this many people like more likely, like less or more in terms of that SNP? Like what are like some numbers there?
I don't know that either of the p value here or the OR value give us a percentage-based thing. You guys have any idea about that? But I think the fact that the negative log base 10, the p-value here is pretty high for that SNP, and then also the OR value is, in the grand scheme of things, not super far from 1, but like, a few decimal places away from 1, which also a 1 OR value indicates no association, shows that there definitely is an association, but yeah, we don't really know like the percent or anything, but it was definitely notable. Mia.
What are the implications of other people knowing your DNA bank data?
Yeah, so that's another thing. So you could find out something that might change your perception of yourself. But another thing that's interesting to look at is, so 23andMe does have all of this data, and any company would probably pay a lot of money to get their hands on that data. And I think if you look at the privacy policy for 23andMe, they're not going to sell your data. They're not just going to give it out. But in the future, that could be something that like companies are looking for, because this is a super like interesting and also like plentiful source of data that could tell you a lot about a person, because their genome tells you, a lot about them.
Can I just tell you guys about a weird SNP? So there's a SNP that is on the monoamine oxidase gene that predicts criminal behavior. So if a person with this SNP is raised in a household with violence, they have a 50% likelihood of being arrested for a crime.
And that was probably determined through a GWAS study, right?
That was determined through GWAS. So like this, it's important to think about who owns this data, who has it. Should 23andMe even be able to publish data with your genome, or should it like belong completely to you? I'm open and curious here. Sorry, I'm taking
Yeah, no, but does anyone know of any other like cool SNPs or anything that like maybe you wouldn't want 23andMe to have like knowledge of, or anyone? Micah?
When I'm thinking of like what knowledge they have, if they are publishing, do they have the data connected with my name? Like I know for a lot of other private things, like Apple might collect all of your Hey Siri data, but I believe that they don't keep all of your audio recordings next to your Apple ID. I believe that they have that for big data stuff, but not specifically tied back to you. Do you know the status of that with programs like 23andMe?
I definitely don't think that they're sharing it with like an attachment to your name as of right now, and I'm sure you could look at like the privacy policy and read that thoroughly, and find out the like direct answer to that. But yeah, that's a good question.
I can answer that.
Yeah.
So in this case, all the data is contained in a single cell. So if you think about the amount of space that this thing is taking up, it's tiny, and yes, that is associated with your name in their storage bank.
And also your genes, if you like, or your genome, the information in your genome is being used to conduct these genome-wide association studies. So that's another thing to think about.
Do you know the race makeup of like everyone with the data through like the 23andMe?
Yeah, so this is just the data that they used. Obviously they weren't using all of their 12 million customers to conduct this GWAS study. I think it was something around like a few, like 100,000, maybe a few tens of thousands. And they didn't have a ton of data for any of the ethnic groups that aren't listed here. These are the three that they had the most data for. But that's another interesting thing to look at, because for this specific study, as you can see, the meta-analysis and then the different ethnic groups all being kind of within the same range, the like what racial or ethnic group that they were in didn't really matter that much for this specific SNP, but it definitely could for some of the other ones, which is also interesting, yeah.
You mentioned talking about how knowing about a SNP that might cause a likelihood. I remember reading an article or watching a video about something related to 23andMe publicly about publishing SNPs about Alzheimer's, and that got them a huge lawsuit.
Yeah.
Bring that up.
Also, I feel like another implication is like regardless of whether it's attached to your actual name, like if the criminal one is found out to be like associated with something like race or something like that, that can have really big consequences.
Totally. Yeah. Yeah, so the question was, first of all, are these GWAS studies using a lot of self-reported data? And the answer for that would be, I think yes, if not always. This one obviously used a lot of self-reported data. People are reporting whether or not they tested positive for COVID during the pandemic. But I think to connect like a gene to anything like observable in the world, you would have to have that thing that's observable be self-reported by the user, which obviously might skew the data, as well as Juliet's other question, which was, what's the like socioeconomic implications of the fact that, or like the data implications of the fact that people of only a certain socioeconomic class might have access to these tests. So again, like with this one, it didn't skew the data that much, because as you can see, just along all human populations, it's pretty consistent. But yeah, with certain data that we don't have, like, or with certain SNPs or certain pieces of information that we don't have data for everyone, that might skew the data a lot.
Can you imagine what would happen if Facebook got a hold of your 23andMe
Yeah.
data? Like if they could correlate that behavioral data set with your S&P analysis?
I mean, the ads was like, oh, I'd be so happy about that. Like everything, oh my gosh, this is perfect ad.
They'd be like, do you have this very specific disease?
Yeah.
Like, yes, I do. How did you know that? Like, why did you know that? Yeah.
So it'd be like people that had like high anxiety, their ads would start like, you're about to die.
To, like, get
Buy
like,
this
like,
soap,
yeah.
right? Or like, it would be, yeah, they're not going to give you what you want. They're going to give you what they want in a way that you know, they know you will buy it.
Yeah.
People like higher propensity.
Because like your search history and stuff.
Just like alcoholism.
Your search history and everything you do on the internet is already giving you very specific ads. And I'm sure, yeah, that tells you a lot about yourself, but the genome, that has the potential to tell anyone like so much more about you. Micah?
I real quick looked up the price. It's about $200, give or take a few, for like full report. There are somewhat cheaper ones, but that's their standard price. And so, while that's not prohibitively expensive as it once was, like there's a lot of people who can't spend $200 for something that doesn't have like a direct application for something of use. And you can see that you can see these sorts of inequalities reflected a lot more European people, I would guess.
Yeah, you're right, because
and so the error bars are a whole lot smaller.
yeah, exactly. For anyone who wasn't, I think Micah picked up on the error bars, but pretty much the vertical lines that are associated with the dot, I guess a smaller one would indicate less error, meaning that there was a larger sample size for the European ancestry group. That's where they had the most data, so there's the smallest